What if I told you that most of the animal research we fund in 2025 wasn't just cruel, but useless? Keep listening as we uncover how antiquated animal experience wastes billions of dollars, delay medical progress, and keep better science on the sidelines, and what you can do to make a difference. Welcome to Puppies, Pandemics, and Public Health. I am Dr. Johnny Lieberman. If you've ever wondered how your everyday choices, including what you eat, what you buy, what you wear, and the medicines you take, impact your health, the health of your family, animals, and the planet, you are in the right place. Let's get to it. Dr. Zaher Nahle, the CEO of iVictory Solutions, a market research company specializing in the biomedical discovery markets, is also a national leader in advancing cutting-edge biomedical research, including novel alternative methods, or NAMs. He also serves as the Senior Scientific Advisor for the Center for a Humane Economy and Animal Wellness Action. Dr. Nahle received his PhD in physiology and biophysics from Stony Brook University, Cold Spring Harbor Laboratory, and an MPA from Harvard University. Hello, it's Dr. Johnny Lieberman. I am here with Dr. Zaher Nahle, and this is Puppies, Pandemics, and Public Health. Dr. Nahle, thank you so much for being here today.
Thank you, Dr. Lieberman, and congratulations on this incredible podcast.
Thank you. Appreciate that. I'd like you to start with the most important topic. If there was one issue or one message you'd like to make sure that our listening audience did not miss throughout our conversation today, what would that be?
The key message, I would say, would be to be engaged. If we are talking about medical progress, drug development, innovation, at the end of the day, it's your taxpayers' money that makes the difference. So your engagement is very important, that you take the time occasionally to write to your lawmaker, express your views on these issues, on the medical issues, so that they can hear from you. You have no idea how important that public engagement is directly from constituents. So we are going to discuss a variety of things, and we are living in an exciting time, quite frankly, that we have seen recent progress on bolstering innovation away from animal testing, using innovative technologies that are poised really to transform the way we have conducted drug development and basic sciences. But at the end of the day, it is essential for people to be engaged. You don't have to be knowledgeable about the subject matter too much, but just express your opinion and reach out. I believe that's one of the messages that I have for everyone, for myself too. And I'm guilty sometimes at not doing that often, but we have to do that.
Thank you. I can't emphasize that point enough. We all have the power. If you can vote, you have a say in what your lawmaker does. You have a say in where your taxpayer dollars go, what the government invests it in. If you choose to say nothing, that's your choice, but you have the power to do so. And your lawmakers are bound to at least listen. Everybody has something going on, but the issues that are most important to me, I want to make sure my voice is heard with my elected officials. Zaher, why don't you share a little bit about your background? We're going to talk about animals and research for drugs for human use. And Zaher has a special, unique experience in this area. Perhaps share a little bit with our listeners so that they know exactly how qualified you are.
Thank you, Johnny. I do not want to bore people to death. Let me be very brief in terms of my background. I'm a scientist by training. I received my PhD through a joint program between Cold Spring Harbor Lab and Stony Brook University, that's in New York. Then I did a postdoctoral training, and then I got my own lab at Weill Cornell and Vanderbilt. My PhD was in physiology and biophysics. Then I transitioned to the Harvard Kennedy School, fellowship in public policy and management, and also did a master's degree also at Harvard in public administration. Now I have my own market research company, and one of the titles I'm the most proud of is my association with the Center for Humane Economy and Animal Wellness Action. These are organizations that are promoting innovation and animal welfare and really trying to create healthier, compassionate societies. They were the ones that really shepherd the FDA Modernization Act that we may get to talk about during our conversation. And I'm their senior scientific advisor for issues like that. But one of the things that I'm proud of is really my work on driving technology and innovation to replace animal testing, because this is something that will affect every single one of us. Anyone who is getting to an age to take medicine just to keep going, or have a patient suffering from a rare disease or a common disease like cancer or heart disease or infectious type diseases, everyone is going to be touched by the shift from this bizarre reliance, Dr. Lieberman, on the obsession, I would say, with testing on animals that have yielded 95% failure rate across disease domains. I mean, imagine that number, 95% of clinical trials, 92 on average across all diseases, fail for new drugs, although these drugs receive perfect safety and efficacy profile in animal testing. So that is a very concerning business model that has been operating for 100 years or so in the United States. And now we have technologies that can reduce that failure rate and that outperform the antiquated animal testing. And agencies started to pay attention and develop shift in their policies. We just started with this administration in April 2025. So this is an exciting time for a new phase, for a new era in drug development and science in general, biomedical sciences.
Thank you for expanding on that. And just to be clear for our listeners, you actually worked in a lab testing on animals, working to develop drugs for humans. Is that correct?
Oh, yeah. I had a very large program at the time. We had programs on cancer, we had programs on cardiovascular diseases. And yes, we are able to cure cancer in mice. We certainly eliminate heart disease in rodents and in large animals. But when it comes to translating that into the clinic, this is where the issue is. Because one thing we miss to realize and have missed to realize for a long time, that's how our training, whether you go to graduate school or medical school, it's biased towards research on animals. We fail to realize that there are radical differences, whether immunological, pathological, genetic, anatomical, molecular, pathological differences between animals and humans. We were discussing the futility of focusing on animal testing for a long time with unacceptable, really indefensible failure rate when it comes to translating to the clinic. And you asked me about my lab and how we used to do animal models. Certainly, we and others could cure all type of diseases in labs, but the issue is with translating that into the human system. This is where the failure rates are happening. And as you know, Dr. Lieberman, this squandered billions of dollars, irrecuperable time in terms of talent wasted, and really very costly delays in terms of medicine reaching patients.
What made you decide to leave and switch paths?
Now, I'm always fascinated by health policy in general. I knew there's a better way of doing things. And I was at a point in my life where I was very attracted to trying to contribute to systemic changes at the policy level, because I understood that if you can change those bottlenecks, you can benefit not only your lab or your department or your university, but actually you can benefit humanity directly if you have the right policies. And that's a passion of mine. I have that inclination, not only scientific, but also in terms of policies and administration for nonprofit and for public. I thought I'll combine both. I didn't know I'll end up here eventually. I was very excited at trying to combine the technical knowledge with better understanding of policy and how legislations are made and how our government administer its plan for developing drugs and for conducting basic sciences.
I found that that's in general very difficult for anyone to do who's been on one career pathway, trained to do a certain subset of things. You're getting a paycheck to continue doing that. However, I just want to grasp on to one of the words that you said, futility. As everyone who watches this knows, and listen, I practiced medicine for over 20 years. I'm on the extreme end of the research pathway, from even before animal testing to getting to humans. And I can look at research studies when we may have 10 years of data showing that the model is not accurate. It's not able to predict or translate, like Zaher was saying, it doesn't translate to humans. So why do you keep funding it? Why are you spending my taxpayer dollars to keep funding an experiment in futility?
That's sad and maddening at the same time. I know a lot of people who work for the federal government at the NIH and the CDC and the FDA. And these are some of the most dedicated people in general in our federal government. But there are several factors to this, as you know. But outside interference, political pressure, groupthink, work avoidance, kicking the can down the road, money. Let's not forget that most of the folks who are on study committees, NIH doesn't review grants really. NIH asks academics from universities to come together and review grants. Most of these folks are animal modelers themselves. They use animals in their lab and are trained by people who taught them how to do that. So this is a propagation of bias. And those people wield a lot of power at the NIH. Of course, not all of them are sympathizers with animal experimentations, but a large number of them are. So that's part of the issue. Historically, if you want to go back to 1938, when the Food and Drug and Cosmetic Act of 1938, which established the FDA, number one, and gave it enormous authority, but also established the requirement for using animals to test for toxicity. Although the animals were never validated to be tested for toxicity, that happened as a reaction to a scandal at the time. A toxic chemical was given to people and 100 people died. So as a reactionary policy, that requirement was established. Fast forward to 1962, another unfortunate event happened with pregnant women. They were given drugs for morning sickness, and then that drug turned out to be causing birth defects.
This is thalidomide.
Yes. So as a reaction to that, they were tagged on the animals to test for efficacy. Again, in animals, even though animals were not validated to determine efficacy. In fact, many clinical trials failed, 95% of them, precisely because of that mismatch in modeling and other reasons. So if you look at our history in terms of why animal testing developed, it was the easiest thing we could grab, the best thing to quash public outcry in response to disasters and give the public a false sense of security that, look, we did something about it. And of course, understanding that at the time, we didn't have the sophistication in technologies, we didn't have the molecular biology knowledge that enabled us to distinguish between the really molecular characteristics of drug development between animals and humans. But now we do. Now we understand more. Now we know more. So it's inexcusable, and it has been inexcusable for a decade or so, to keep this focus on animal testing without looking seriously at alternatives. And luckily, that is changing now.
I'm glad you brought all that up. A dominant discussion now about validation of the non-animal methods. But I think, as you just brought up, I think most, if not all, of the animal models that we use today and historically, they haven't been validated either, correct?
Correct. Absolutely. So there are two types of testing, right? So sometimes you test on healthy animals and sometimes you test on disease. So these are mice or dogs or non-human primates that we give them a disease and we hope that this disease looks like the disease that happened in human, whether it's a cancer or diabetes or heart disease. But the reality, most of these models do not even resemble the cancers that happen in humans. I can give many examples. But what I want to mention also is, for example, in the testing of monoclonal antibodies, these are agents, these are important factors in treating a variety of diseases. They have been around for 20 years, so we know a lot about them. There is a history of using these monoclonal antibodies, as you very well know. And FDA just recently terminated all testing on monoclonal antibody development in animals, replacing them with what we call NAMs, new alternative methods or new approach methodologies or non-animal model, whatever you like to call them. And these are artificial intelligence modeling or what we call organ on a chip, which are these miniature live cells and tissues that resemble the biological context of humans or organoids. So that's an important decision by the FDA to eliminate toxicity testing in a major class. So step after another, we are now seeing the speechheads of NAMs replacing and outperforming traditional animal models in the safety testing for chemicals and for drugs. It is really very promising to see such a thing.
So right now, the NIH budget for research is around 40 billion, if I'm in the ballpark. And a sliver of that, I think it's less than 1%, is for NAMs. What if, for example, that budget were flipped and we said, we think this is a priority? As you've discussed, animal models are largely futile. They fail repeatedly. We're not only wasting money, we are wasting time. We're wasting time where we have these smart researchers maybe devoting 10 years of their life to an outdated mouse model, which has almost a 0% chance of producing something, when if we gave them, I don't know, $10 million and say, let's go do this with NAMs, might not be able to develop more quickly. Let's get treatments to patients more quickly. That should be the driving factor as a clinician working on the back end. I want another treatment more quickly. And why are you relying on a method which is shown to have an extremely high failure rate? It abuses and tortures animals with a very low yield, and it's wasting scientists, brilliant scientists' time. Is there a way to somehow flip the script on that and advance this?
I was just reviewing an editorial that is almost accepted now with revision in the NAM journals. And actually, I raised exactly that point about funding. There are 6 recommendations to the NIH leadership. One of them, I don't think I'll be scooping anything if I just mentioned one of them. One of the recommendations is redirect 40 to 60% of funding allocated for animal testing to human relevant NAMs. So a recent Government Accountability Office report in 2023 estimated that the amount the NIH spent on animal models is 5.5 billion annually. Now, many, including myself, believe that this is an underestimation. Some believe it's up to 20, 25 billion. However, let's take the GAO number as a start. It's a good start. And by that estimation, Dr. Lieberman, we are talking about an investment in the range of 2.2 billion to 3.3 billion annually in NAMs. And the key here that I note in these suggestions or recommendations is that that money should not be new money. Because if we get new money and we pump it into NAMs, but not redirect that money from already existing budgets for animal models. We really, maybe in the long run, in 100 years, yes, you can be somewhere, but as long as you keep pumping enormous amount of money into animal testing and into animal modeling that eventually fails, we are not going to be in a reasonable amount of time be able to reach what we want to reach from NAM, because they will always be diluted. As you said, the expenditure on NAM is infinitesimal compared to what's spent on animal models and on animal experimentation. It's less than 1%. The NIH is rolling out certain prizes and initiatives, but again, they are ranging in the 7 million to 80 million, which is a drop in the bucket when you are talking about transforming a field and operating on a 50, 60-year deficit in terms of investing in NAMs. So it's very important for the NIH leadership to really realize that they are operating under this urgency.
And the urgency that you mentioned, I find that it could be a key component for helping mobilize the public. Perhaps you or a family member of my listeners, are you suffering from a so-called rare disease that pharmaceutical companies are not investing in because there's not profit to be had from it? Or are you suffering from a disease that perhaps the treatments aren't great for, or that there's a significant side effect profile which may have been discovered only after it entered human trials? Patients are suffering needlessly as a result of this antiquated system of research, which produces products which have unintended consequences and don't work as well once they get into humans. What if we took that chunk of money? We took those scientists that are, in essence, wasting their time on a model that's almost certainly not going to produce anything. What if we took them all and said, here's a chunk. You've got a greater chance of finding a medication doing this. Let's do it. Let's get something out to these patients who are suffering with these diseases more quickly. One of the ways that I think about it, you've heard the expression, even a blind squirrel finds an acorn once in a while. That's the way I view using animals in research for finding drugs in humans. Does it happen? Sure. Is it the most efficient way? I think, Zaher, you can testify it's by far not the most efficient way.
It's absolutely not. We are saying now is there are better technologies and approaches that we need to start incorporating into our quest for the truth and our investigation. In a personal conversation with Francis Collins at one point, and I think he used to say that in public.
Former NIH director, correct, for our audience?
Correct. He told me that it's very difficult to manage scientists. Managing scientists is like herding cats. One thing you can do is move where you put their food. And although this is a crude analysis, I think that you can direct policies and make a group of people who are opinionated follow a certain path if you provide them with the resources they need. We expect from Dr. Bhattacharya and his leadership to recognize that there is an urgency. Again, the 95% of the rare diseases that do not have one single FDA approved drugs, there are people behind these, there are suffering behind these. The 95% failure rate in oncology, the 96% failure rate in neurology, these are unacceptable numbers in common diseases that you and I or people we know have. So there's the urgency that the new director has to think about here, because he's the man entrusted in medical direction of the nation. And this is a position of honor. This is a position that serves humanity. We look for leadership to change the status quo, and he's the one who can do it.
So you made a transition. You felt your work was futile at some point. How common do you think that feeling is among those who spend all day experimenting on animals for a government paycheck? And why are there not more of them who probably have a similar, if not identical, understanding of it? Why are there not more saying, hey, we don't want to do this anymore. We want to move to NAMs or some other method that's going to actually be more impactful?
Well, they are stuck in a vortex. They are almost shamed in labs if you don't embrace this in vivo, which is considered a superior level of work when you submit your paper from your lab to do in vivo experiments because it's almost automatic. If you don't have in vivo experimentation in a paper that you submit, that the reviewer asks you to do this in mice or to do this in rats or to show him or her that this is also valid in vivo. And this is where I first started feeling the pressure that it's inescapable for you as a scientist who's trying to publish in high profile journals not to do animal models. Pressure by the community, by the publisher, by the reviewers to ask them for these kind of experiments. And that also must change. And I know there are lots of people who are trying to change that mindset in terms of in the publication sphere and the reviewer bias.
Thank you. And also, you talked about bias. I wanted to bring up another type of bias inherent in a lot of this literature, which I don't see talked about very much. And I want to emphasize this in terms of the futility. So the number that was brought up by Dr. Nahle, 95% is what you'll see published in terms of a failure rate for medications that get passed through animals and then fail in human trials. There's something called publication bias. And it turns out publication bias is something where in publication of research, not even just animal research or medical research, there's a bias towards articles which have a positive result getting published over articles which have a negative result. And this applies in this area. And the number that I found, which is approximately 1 to 5 in the animal research space. So for every one positive result you see in, say, mice sepsis or mice model, there's probably 5 studies looking at the same thing, which showed a negative result, and you're never going to hear about them.
And hearing about these studies is so important because you can save enormous amount of time and talent by just looking at the negative results and avoid the pitfalls or try to correct them or abandon that project before it's too late or before the investment eats up your time and money and will. So that bias exists towards only publishing positive results. And I think pharmaceutical companies do the same thing too. The industry is not immune from that. But there's also another type, 89% of publications are not reproducible.
Can you just explain for our listeners, what does that mean? 89% of studies are not reproducible.
They published studies saying that inflammation causes breast cancer. Use the same technique, even ask for the same material, but got different results or could not reproduce what Nahle lab produced. Another lab in Canada did the same thing, could not produce. Another lab in Netherlands did the same thing and could not produce. But these papers get published.
In this example, maybe only your paper gets published with a positive result. Mine and the Canada lab don't. So it looks like you've discovered something great, which may move forward, ignoring the negative results.
Correct.
So here we've touched on so many important topics. You've touched on the antiquated nature of current state of animal research, the high failure rate in animal research, the clinical futility, the potential to redirect funds to help get these treatments out to patients more quickly that need them. What can our listeners do? What are a couple of steps that they can take? Let's teach our listeners 1 or even 2 things, decisions they can make differently, people they can speak to. If any of this resonates with them, how can they make a difference?
They can make a difference by following an organization like the Center for Humane Economy and call or email any other organization that they feel represent them on these issues and email their representative, call their representative, make their voice heard about these particular issues because there are thousands of bills every year. And there are a few of them that are trying to change the status quo and improve the drug development space and improve animal welfare. So if any of these topics move you and shake you and you want to spend 2 minutes just voicing your opinion, this will go a long way. And actually it can change. Participate, pick up the phone, call, email, text, WhatsApp, anything, but make your voice heard when it comes to these important issues about the future of drug development, about the future of animal testing, about your prescription drug cost, and about your overall cost of healthcare. And by doing that, you actually will be helping, you will be driving forward and exemplify your pride in your nation.
Thank you. I couldn't echo that strongly enough. And so much of, if not everything of what we talked about, you can probably find out there. We live in a world of transparency. So something that we talked about touches you. If you have a family member who's been diagnosed with cancer, or you or someone you know has been treated for sepsis and maybe didn't make it or had a bad outcome, look into these animal models. Researchers are still doing some of these models and the government is still funding them. And that's really why the researchers are still doing it in large part, even though they fail time and time again. Don't you owe it to yourself and anyone you know who's suffering from medical conditions to speak up and try and ask your taxpayer dollars to go somewhere that has a high probability of leading to a cure more quickly? Let's invest the time and money that we have on human-centered methods, human-relevant models, models that are translatable, that have a high probability of getting a medication that's going to be safe and effective in humans. If something I said speaks to you, please look into it, educate yourself, and take action as Zaher just mentioned now. Dr. Nahle, thank you so much for your time. I appreciate and value your opinions and all the work you're doing with Center for Humane Economy to advance this important area. Thank you.
Thank you so much. And again, wonderful to see you and to hear your thoughts. And congrats on this very informative podcast. And we are in this together. So onwards.
Thank you. Thanks for listening. These conversations aren't always easy, but they're important and worth having. Take what resonates, ask questions, and keep learning. Find us on Doctor Podcast Network or wherever you get your podcasts. The content of this podcast is for informational and educational purposes only. It does not constitute medical advice nor legal advice, and it cannot replace professional consultation with your doctor, lawyer, or other licensed expert. Always seek guidance that's specific to your situation.